Types of database logs in clinical trials. Introduction. Now, in clinical trials, database logs are critical milestones that ensure data integrity and analysis readiness. They are used to prevent any changes to the clinical database once the data is entered and considered final for a specific purpose. For example, say interim or final analysis. Now, today I'll be discussing about different types of database logs and their importance, implementation, and what are the differences between various types of database logs in clinical trials. Type 1 soft log, interim log. Now this specific log is a temporary one where the database is frozen for an interim analysis or review, and it can be reopened whenever required and whenever it is needed, provided you have given the specific reason and the rational reason, right? So now when it is used, now it is used before interim analysis that needs to be conducted. So say for example, I want to conduct the interim analysis, right, for that specific study. So what happens is, before conducting that interim analysis or before performing that activity, the database has to be locked temporarily, right? So it is locked temporarily and it can be reopened after the interim analysis is submitted or whenever it is reviewed, and whenever any new patient's data come, when you will require to enter those certain database. Now this kind of interim analysis is conducted just to ensure that whether the clinical trial study is going on the right track. Now say for example, after conducting the interim analysis, it so happens like the result are not appropriate and the result is not up to the mark and the safety and efficacy is not meeting certain parameters or the hypothesis that it is met, right? So what happens, like in rare to rare cases, if the sponsor decides to stop that clinical trial based on the stopping rules criteria which are available while conducting the interim analysis. So whenever an interim analysis is done, there are certain stopping rules criteria so that you can stop the clinical trial if it is not appropriate and the clinical trial is not going in the right direction and there is no benefit to the patients and there is no improvement related to that disease after consuming that treatment. So this is a very important kind of an analysis, and generally this temporary lock is done before the interim analysis is started, right? Now DSMB is the data safety monitoring board. Now again, this is a very important and a crucial step. Now whenever there is a data safety monitoring board review happens, so what happens is, especially those safety domains which are labs, adverse events, medical history, concomitant medication, vital signs. So these parameters are usually summarized and they are tested whether we are having sufficient data, whether we are meeting certain criteria, right? So whether it is going to be affect the results or we are going to get the benefit from this treatment, or whether the treatment is doing good or performing well or not. So that kind of decision is made in the DSMB review. Now say for example, if a medicine or a molecule, it so happens like it is not going in the right direction. Now there are so many serious adverse events reported in the DSMB criteria, right? And it is affecting the health of the human subjects. So what happens in this scenario, there is always a possibility of an interim analysis or the DSMB review. You can actually stop the clinical trial by certain stopping rule criteria that have been met. Now those criteria, they come at the time of the DSMB review and the interpretation of the results, if they are not satisfactory and if they are not sufficient enough to go ahead for that clinical trial, right? So this is a very important kind of a step, or wherever there is any data review meetings, whatever happens with all the stakeholders, in such scenarios certain decisions are taken. Hence this particular type 1 is a soft lock and an interim lock, it has been told. Now can it be modified? Yes. Now this specific lock, it can be reopened provided you have the valid reason. Now say at the time of the interim analysis you temporarily locked it. Now after that you conduct the interim analysis and the DSMB review, right? So if the stakeholders and the sponsor is giving a go ahead to continue with that clinical trial, then well you have to unlock this specific soft lock, and you need to unlock it, and then again you need to enter the patient's data, and then you can start continuing working on your database as usual in your work. Now the type 2 is the hard lock, which is the final lock. Now this specific lock is a permanent lock after all data cleaning and reconciliation are completed and there are no further changes allowed. Now say for example before conducting this final lock or the hard lock, what happens is whatever there are queries which are raised to the sites, all the queries they needs to be resolved, everything has to be reconciled, reporting adverse events and asking any questions related to any kind of data issues or laboratory vendors issue. So all those changes have been cleaned, reconciled, and there are no further changes needs to be done. So this is a specific hard lock which is the final lock, right? Now when it is used, now it is used before conducting the final statistical analysis and clinical study report. Now here I'm talking about the final. Now what happens is as and when the database it is started designed and it has started entering the one or two patients subject data, minimum two patient subject data should be there, so that the programmers and the statisticians they can start doing the programming activities, and they can start preparing their code or the program ready. Now that is the draft version, right? So draft version statistical analysis can be done before the hard lock, right? So that much sufficient time the programmers and the biostatistician they get in terms of keeping their codes ready. So now once the hard lock and the final lock is happened, and where all the changes have been completed and there are no further issues present in the database, then at the final statistical analysis you just need to rerun, and you just need to check whether whatever the issues that were raised before the hard lock, all those have been resolved and it is not impacting the statistical analysis. Now say for example after this hard lock the final analysis has started. Now during the statistical analysis, which is the final version, it so happens like the statistician or a SAS programmer they come to know a specific data value was not updated, right? Now it's a very sensitive matter. In these scenarios it's very essential that biostatistician and programmer have to double check the value whether it was correctly done or not done, right? So if it was not done and if it's a mistake at the data management side, then again it needs to be informed to the sponsor, all the stakeholders, and like you need to go to proper approval process and you need to go to proper ethics committee, and all the stakeholders they need to be involved so that you can make the changes. Now this happens in very rare to rare cases. If any such kind of unlocking situation at the time of the hard lock it happens, that means the team did not did their job properly and there was performance issue and the quality issue, they did not monitor things and they did not check the data issues value. Now these kind of situations happen in very rare to rare cases. Now if the team is performing well, they are checking all the issues properly, they are ensuring that all the changes that were supposed to be updated in the database, everything has been done and there are no issues and they are 100% confident to it, then the hard lock and the final lock it happens. And if there are no changes at the time of the final statistical analysis and the programmers and biostatistician they feel that everything is going well ahead, then there are no changes are allowed, and respectively you cannot make any modifications once the hard lock happens to that specific database. But in very rare to rare cases 1% chance will be there where the team did their job properly, so hence there might be some situation you might have to unlock. So that's not good for the clinical trial study, and again that needs to be reported to the FDA and the European regulatory agency as to why did you unlock it after hard lock, because that is usually not expected and that is not the procedure that needs to be followed in the clinical trial, right? Hard lock is the permanent. After that, there are no changes allowed in the specific database to make any changes, right? Now another type 3 is the snapshot lock, which is the freeze or the cutoff. Now what do you mean by cutoff? Cutoff is nothing but a specific point in time where the database is selected for doing the subset of analysis or regulatory submission. So say for example, I have a specific database. Now I want only to select only those records, like say up to March or April, right, to specific time point, or I need the database for first 6 months, right? So it captures the state of the database at specific point in time for a subset analysis or regulatory submission. Now who decides the specific time point in this freeze or the cutoff? It's usually decided by the sponsor and the ethics committee, so as to ensure that what kind of results they are expecting. Now it is used especially for achieving milestones, like say for example reaching 100 patients or database submission. So this kind of a snapshot or the freeze or the cutoff lock is used when you have certain milestones, like say in my database I'm having only 100 patients, so I will do the analysis for that 100 patients, right? After that I will do the snapshot or the freeze or the cutoff. So these kind of situations you can use the specific log. Now can the data be modified? Yes, data may change after the snapshot unless a hard lock follows. So in hard lock it's very unexpected that you cannot make any changes, but whereas in snapshot it's just a cutoff point, like you are just extracting data only to that much time point or this many number of patients. So yes, you can make changes, like because it's not a final hard lock. Now next is the type 4 subject level lock. Now this lock is nothing but locks the database for individual subjects once their visits, queries and reviews are completed. Now this is mostly seen in the oncology clinical trial studies, where you use rolling lock, as and when the subject completes the study. Now say I have a particular subject 001. Now that particular subject, from start of the study, which is screening, then giving the informed consent, then taking the consumption of that treatment dose, then giving the vital signs, lab signs, parameters, and everything is being done. The subject completed the study and he has follow up and everything is okay to it. So there are no further issues, there are all the data is perfect and there are no issues into it. So in those kind of scenarios you can especially lock the subject as well, that is the rolling lock, as each subject completes the study. So you can lock the database subject wise, right? Now yes, you can make changes to the database for others, but not for the locked subject data is frozen. So whatever subjects that you have locked, it's completely freezed, right? And you have freezed only by ensuring that whatever the data is captured is perfect and there are no further changes needs to be done. So in those scenarios you can make changes, but only for the other subjects which are unlocked. Now next is the site level lock. Now this particular type of lock of database is at the site level after all subject data from that site is cleaned and reconciled. So say for example I have a specific clinical trial study which is having 10 sites, right? So at one specific site all the subjects have been entered, all the queries are resolved, there were no essays reported, there were no deaths reported, and everything was working smoothly and there were no issues at all, and there was pretty much good amount of cleaned data from that site. So in those specific scenario you can log that specific site data in the database, and you can work on for the other sites. Now when it is used usually at the site closure or before interim review. Now say for example it so happens like you have multiple sites, like say in specific country you have 10 different sites where the clinical trial study is started. Now it so happens, due to the logistics issue of the management, or due to the budget, a specific site happening in that specific area, they have planned to close it, right? So what happens is, in those scenarios where there are any logistic issues, you can specifically get the queries resolved as soon as possible before the slide is closed, at any point of time. So in those scenarios, you can lock the data by site wise or before any interim review. Now you can make the changes to the database and you can modify it, but other sites data may still be editable, right? So you can make changes to other sites, but other sites data may still be editable. So whatever the site that you have locked, you cannot make any changes to that, but for the other sites which are unlocked, you can make those changes. Why database logs are important? Now these database logs are important because it ensures data integrity. Now just to prevent any unauthorized or any accidental changes, like during the critical analysis stages, it's just that we need to prevent those kind of activities. That's the reason the database is locked. Now say for example, there could be any so many team working in that specific database, right? So if somebody who has the authority or access to that database, they might do any accidental changes. Now once the subject's patient is entered in the database, it is cleaned, everything is proper, the patient has completed the study properly, and the patient has completed the follow-up activities also. Now after doing all this, there could be some changes, like the unauthorized person is, like say somebody from out of the team, or somebody like trying to manipulate the data, or somebody has just accidentally, like working on some another study, but by mistake made changes to the another study, right? So in those kind of scenarios, just to avoid those kind of situations, so that no changes can be done, in those scenarios the database lock is very important to conduct. The next is the supports regulatory compliance. Now the regulatory agencies which are there, FDA and the EMA, they require evidence. Now what is evidence? Evidence is nothing but whatever the human patient's data which is there, and that what you are entering, and those entered data is analyzed, right? So that itself is the evidence. Now so happens, like if the data which is an evidence as per the FDA and the EMA agencies, now if that itself is being manipulated by someone, then but obvious, the integrity will not be there, the honesty will not be there. So in those scenarios, the regulatory agencies, they require the evidence that data should be used in analysis that has not been altered post log. So once the hard lock happens to that specific database, there is no way that anybody from the team, not even the sponsor, can make any changes to that specific database. Now it enables accurate analysis. Now data must be stable during the statistical programming and analysis. Now why it needs to be stable? Because we are performing the final analysis. Now imagine if your database is changing at the production level. Now the analysis also and the programming also, it will affect, to some extent it will affect, right? So in those scenarios, whenever the database, it needs to be stable. Now that stable, it can happen when all the subjects patients, they have entered the database, the subjects have completed the study and they have entered, and now once the database is stable, means once the hard lock has happened, so then the smoothly statistical analysis and programming can be done. Now next is the traceability and audit trail. Now the logs are logged and auditable, showing exactly when the database was finalized for analysis. Now the traceability and audit trail is very important factors when you're working with the database log, right? Integrity, compliance, accuracy in data, then the traceability and audit trail. Now these are all the reasons as to why we conduct the database log. So audit trail also, it plays a very important role, so that you know when it was logged, when it was performed, what all issues there were there, and what are the logged kind of information which is present. Next is the milestone management. Now these logs help meet clinical milestones like say interim analysis, safety reviews and final reporting, right? So even if I'm trying to do the interim log, now remember one thing, before conducting the interim log, temporary the database can be logged. The reason is because even the interim analysis can play a very important and a vital role if it meets the stopping rules criteria for the clinical trial study. So this interim role, if it has that much power to stop the clinical trial if the required results and the satisfactory results are not making, so in those scenarios, that's the reason we perform soft lock and the temporary lock, so that nobody can make any changes, any unauthorized person outside the team or outside the company or any outside making any accidental changes being in the company or being within the team. So those kind of scenarios to help to meet the milestones, these are the important factors as to why do we conduct the database lock and why they are important, especially integrity, compliance, accuracy, traceability, audit trail, and just to meet all the milestones. Now, why are they implemented? Now, they are implemented to formalize the completion of data collection and cleaning. Now, the database lock is something like say for example, very simple example that I will give. You have money, you have gold, you have silver at your home, right? You don't just keep it open at your home. What is it that you do? You keep it in a locker with a safe kind of place where you lock that specific uh, place, and you keep the keys at a very safer place, so that nobody can make any changes or nobody can take away your gold or money. So it's the same way, it's the same common thumb rule that if nobody should play with the data, nobody should manipulate. So in those kind of scenarios, that's the reason we do the database lock. Now it's generally used to formalize the completion, so it's a process formally, the data was collected, it was entered, it was cleaned, and it was maintained properly. Now just to ensure that nobody plays with that data, nobody makes any manipulation to that data, whatever it was collected with the raw data, it's just like to do the final database lock. Now to prepare the final statistical analysis and ensure reproducibility. Now, so say for example, if the database lock does not happen and if somebody else can make any changes, right? So what happens, a statistical analysis also, it will impact, it will keep changing. So just to ensure that once the database is cleaned, you can lock the database, and so that you have a stable kind of statistical analysis, and that's the final analysis one, right? Just to ensure that it is reproducible. Now to protect against bias, especially during unblinding or interim analysis, right? So just to ensure that there no bias kind of scenarios play in that specific database, it's for that reason we implement the database lock. Now it is to communicate readiness across teams, for example, data management, stats, and clinical team. So this database lock is implemented just to ensure that you communicate across all teams, this is all we have cleaned the database, we have done the query resolution, it is everybody clean. So these all are kind of the scenarios as to why do we implement this specific database lock, just to avoid any bias situation and we have an unbiased area. Now there are certain real world examples which I would like to discuss here. Is the subject level locks are applied as each subject completes their last follow-up visit, right? So as and when the subject completes the study and completes the last follow-up visit, and all the queries and all the data issues, they have been resolved, and there are no further questions on that specific subject. So that subject itself you can log, especially that row in most of the domains, right? So that is one part of the real world scenario. The another is the interim analysis is planned, say for example after 100 patients complete the first 6 months of treatment, right? So for the interim analysis anyways you need a soft log, which is a temporary one. Now the snapshot log is taken for a regulatory submission at day 18. So say for example the regulatory submission takes the decision that we want to see the analysis for the first 180 days, right? So in those scenarios your cutoff point is your day 180, right? Next is after all the patients and the sites they complete the study and all the queries are resolved and there are no further questions on that database, and database is cleaned, organized, managed properly. So in only in those scenarios the hard lock, which is a permanent one, is applied before conducting the final analysis and generating the clinical study report. Please like, share and subscribe. Leave a feedback comment mentioning any topic that you want me to make a video. Looking forward for a positive response. Thanks for watching. Please like, share and subscribe. Leave a feedback comment. Many thanks for your support and having great connection.